is basically the identification, the procedure of DLC. DLC stands for differential leukocyte count. So talking about the procedure of the DLC, we have to be sure during the procedure that the spreader, that is the edge spreader, should be smooth and narrow, and area between the tail and the blood of limb where the neutrophil is mostly concentrated in the edges. And we should be using the junction that side for seeing the slide. Now next we shall be talking about the process. So in process, when we have to see at the angle of 45 degree, we have to take the slide against it. So first of all, in a clean slide, we have to add a drop of blood. Then we have to take another slider and bring it at a 45 degree, touch the blood and make a smear. We have to make sure that the slide is clean and the spreader is smooth. Ideal smear is of tongue shape. So here I shall be presenting a rough range of neutrophils, lymphocyte, monocyte, eosinophil and basophil. Basophil is the least reported cells. It's roughly 1 to 2%. So the relative percentage are neutrophil will be roughly 80%, lymphocyte will be somewhere around 18%, monocyte would be 2%, eosinophil and basophil will be 1%, and eosinophil is very low concentration of eosinophil is observed. So over here I have a eosinophil, right? In eosinophil, the main character is headphone-like nucleus to identify, but that is not a sure-like thing to identify. The main thing, it is called eosinophil. So think eosin. This is stands for the pink color that we are using, the hematoxin and eosin. You have two stains, right? H and D. Eosin is the pink color stains. In eosinophil, we have pink granules. These are over here. You can appreciate the pink granules. So that is for eosinophilic stains. You can see the pink granules, right? That is eosinophil. That is the eosinophilic stain. Next, again, I have a specimen of eosinophil. You can appreciate the structure, bilobe nucleus or headphone-like nucleus over here. And I have the eosinophilic granules. Next, I have a neutrophil. So neutrophil is trinuclear. The nuclear is segmented or lobed in multilobed, and over here we can see various. We can see there are no eosinophilic granules present as such in the neutrophil. Over here I have a eosinophil with neutrophil. This one over here is a neutrophil. It has been represented by N. You can see it is trilobe nuclei with no eosinophilic granules. And next over here I have a eosinophil. We'll be indicating it with E. This eosinophil, we can see the presence of eosinophilic granules over here. This is eosinophil. Next, I shall be talking about large lymphocyte and small lymphocyte. Large lymphocyte is much larger than the RBC. The size of RBC is roughly 7.2 microns, the diameter. Large lymphocyte is much greater in size than RBC. It can be appreciated over here. Then I have a small lymphocyte. This thing with small lymphocyte is the nucleus of the small lymphocyte, the diameter of that, and the diameter of RBC. They are same. These two diameters are same. That is how you identify a small lymphocyte and large lymphocyte. These are the distinguishable things. So now next we shall be talking about basophil. The thing is, in basophil, it is less than 1% of the WBCs. The nucleus is U or S shaped, and there are indentations, large dark purple cytoplasmic granules. It helps in immediate inflammatory response. It releases histamine and other molecules during allergic response and parasitic infections. And basically least reported cell. Mostly when doing DLC, we don't even find basophils. But over here we have seen a basophil. This is how the basophil looks. And next, this is all. So I would like to once say about the procedure. So in the procedure, we take a slide and we put it at a 45-degree angle to make the smear. So that's how it's basically done, right? So while doing the smear, we have to make sure that we don't get some very overcrowding or undercrowding. So I had some images of overcrowding and undercrowding. I may not able to get those as of now. So I shall be sharing. So over here we can see a DLC spread. This is a bit too crowded. See, this is too much crowded. This much degree of crowding should be avoided. So whatever is ideal is this degree. This is also crowded. You can see the RBCs are clumped too much. Over here, this is a eosinophil. You can see eosinophilic granules over here. And neutrophil. Even this is crowded. They're not ideal. This is somewhat ideal. This can be found in the head and tail junction. This, the nucleus of this and the size of RBC. So this is small lymphocyte. So this is how we classify what we are doing. So next I shall be talking about the procedure in a short way. So about the procedure, right? In procedure, what we have is I shall be presenting my screen. So okay, so here is the presentation. So over here, you are able to see the procedure. We'll just go over the procedure once. So first of all, we shall be adding over here a drop of blood to this drop of blood. We shall be bringing the slider black and getting it touched and then just spreading it over. While spreading, we shall be getting a uniformly spread structure and we will be getting a smear. Smear is ideal tongue shaped and these are the cells. So with this, I would like to end. Thank you.